Good morning. My name is Mohammad. I'm a PhD student in the Systems and Synthetic Biology Laboratory led by Professor Rajiv Saha at the Chemical and Biomolecular Engineering Department at University of Nebraska-Lincoln. My research involves metabolic modeling and analysis of living organisms, including microbes like the human pathogen Staphylococcus aureus. Thank you for being here for my presentation. In today's talk, I'd like to give you an overview of my recent and ongoing integrated study of staphylococcal metabolism. So I'll start with presenting my new genome-scale metabolic model of Staphylococcus aureus. Then I'll talk about the model curation, validation, and refinements that I have performed. And then I'll mention the currently ongoing study about understanding the regulatory changes in Staphylococcus mutants. Let's start with the model. Genome-scale metabolic models have been successful in capturing the metabolism of microbial species, discovering about their adaptability, pathogenicity, and also generating new hypotheses about the effect of effective antimicrobial treatments. Staphylococcus is a serious human pathogen that causes a number of diseases in the human body. One interesting aspect about staphylococcal metabolism is that it is highly antibiotic resistant. That means conventional drugs keep failing against it, and there is a serious need for developing new therapeutics. As part of that mission, I recently developed a new genome-scale metabolic model of Staphylococcus aureus in collaboration with Professor Thomas at University of Nebraska Medical Center, and it was recently published in Infeasible Systems Biology and Applications. To do that, I assembled the initial reaction sets from the previously published genome-scale metabolic models of Staphylococcus aureus. I went back to the biochemical databases like AIChE and NCBI to include missing annotations. Then I did a balance check on each and every reaction of the model. Then I ensured that the model is consistent in terms of thermodynamics and energy production. I used a framework called ProMatch to reconcile the experimentally observed and model predicted gene essentiality. And then I finally used different types of regulatory information to further refine the model. I'll go over the model refinement steps in this talk. One important part of metabolic model development is ensuring that each and every reaction in the model is elementally and charge balanced. This guarantees that the model predictions are biologically significant. To fix any imbalances, I added or removed atoms or molecules or fixed stoichiometry on either side of the metabolic reactions. Another important aspect of model curation is the elimination of thermodynamically infeasible cycles. These cycles are formed when a closed loop can operate without any external influx of metabolite. These are thermodynamically infeasible and therefore needs to be corrected to get a sensible metabolic profile. There can be different cases of thermodynamically infeasible cycles. The first case is the presence of duplicate reactions in the model. For example, here you can see the same metabolic conversion done by two different reactions where one is irreversible and the other is reversible. If we have free energy change information, we can then correctly fix the reaction direction, or if we do not have this information, we can turn off the irreversible one and keep the reversible reaction so that this cycle is essentially broken. The second case is the presence of lumped reaction in the model, where we have the same net metabolic conversion but eliminating the intermediate steps. In this case, we just eliminate the lumped step and that breaks the cycle. Another case can appear when there are same metabolic conversion with different cofactors like this. If we had cofactor specific information, which we have for this case, then we can eliminate the other reaction and keep the highly specific one. These are the different ways we can fix thermodynamically infeasible cycles. The next step was to find the gene and reaction essentiality in the model and recapitulate that with the help of experimental data. The Nebraska Transposon Mutant Library, or NTML, consists of the phenotypic screens of about 1900 transposon mutants in Staphylococcus, which is a valuable resource for performing high-throughput phenotypic experiments. Our growth evolution experiment revealed varying degrees of growth inhibition of the NTML mutants compared to the wild-type strain, and all identified subtle disagreements in gene essentiality predictions with other studies. Therefore, the true set of essential genes required further scrutiny, which is why As a conservative estimate, we used a consensus set of essential genes by utilizing the existing knowledge base and also our own experimental findings. Genes reported to be essential in any sources were considered essential unless there was evidence suggesting otherwise. There were three types of positive evidence. First, mutants obtained from Nebraska's transposon mutant library were not considered essential unless it was found to be domain essential. This is because the transposon may have inserted a non-essential part of the gene, allowing a partially functional protein to be formed. Second, if the gene was found to be essential only at high temperature, then it is evident that the gene was incorrectly found to be essential in literature because of a high temperature plasmid curing step in the process. Third, if the gene was found to be essential using a promoter-less transposon insert but not with a promoter-containing method, then the gene is upstream of an essential gene, and other sources found it to be essential due to the polar effect that disrupt gene expression. Therefore, finally, for the modeling purposes, only 167 metabolic genes present in the model were considered to be the core set of essential genes in the current study. Once we had this consensus set of experimentally validated essential genes, we identified the essential genes through our model predicted growth estimations, and then compared the growth and no growth estimations for each of the gene knockouts with experimental observation and classified the genes in 4 different types. We identified 2 different types of inconsistencies. One is called NGG, where the model is predicting a gene to be essential, but it is not actually essential. That means the model is missing some functionalities that should be present. The other type of inconsistency is GNG, where the model is predicting a gene to be non-essential, where in reality it is an essential gene. It can happen if there are erroneous pathways present in the model. To reconcile these inconsistencies, we used a procedure called ChroMatch that tries to add missing functionalities and suppress inconsistent functionalities while avoiding change in already consistent predictions. After applying this procedure, you can see that we reduce the number of the 2 types of inconsistencies, increase the specificity and sensitivity of one model, and also decrease the false variability rate. In this figure, the map of central metabolism in Staphylococcus, where I have highlighted the reactions in different colors to denote addition, change, or removal during the model refinement process. I removed some of the reactions to eliminate duplicates, or when there was no evidence of a reaction to be present, or when we had the correct cofactor specificity information for a particular metabolic conversion. I also changed the direction of some reactions when we had free energy change information or to fix any thermodynamic infeasibilities. With new annotation information and to complete some known pathways in Staphylococcus, I also added a few reactions in the model. We also had metabolite excretion data for a number of mutants in chemically defined mediums, both with and without glucose. So I used some mutant-specific and medium-specific regulations to further refine the model. Upon all these refinements, we obtained the new genome-scale metabolic model of Staphylococcus aureus, which I call AIChE 863. This has 863 genes, 1,545 reactions, and 1,379 unique metabolites. One highlight is that we ensured that there are no imbalances and very few thermodynamically infeasible reactions in the model. This is a diagram of the pathway distribution of the reactions in the model, which is typical of any bacterial genome-scale metabolic model. So this new genome-scale metabolic model that is thermodynamically consistent and balanced will allow us to generate new hypotheses about therapeutics targeted against Staphylococcus aureus. At the end, I assessed the growth prediction on 69 different carbon sources that were experimentally tested, and you can see that my model can predict the true positives and true negatives very well compared to very few numbers of false negatives and positives. After we have a curated and refined new genome-scale metabolic model, we attempted to reveal the metabolic adaptability of Staphylococcus aureus when it is subjected to genetic perturbations. To this end, we performed a metabolic analysis of 8 Staphylococcus aureus mutants in the central metabolism and measured 53 metabolites in different growth conditions in both exponential and post-exponential phases. From these samples, we performed metabolite concentration measurements through GCMS and LCMS, and then identified the differential metabolite profile for the mutants. These results were then imposed on the model to estimate the modulation of flux profile for different mutants. Going forward, I'm planning to figure out how metabolic regulations come into play in different mutants in Staphylococcus aureus when they try to survive. To this end, I'll use this optimization framework to find the minimal regulatory points in metabolism that successfully simulates the mutant phenotype. So we'll try to minimize the number of regulatory effects in the model, while we'll try to capture the mutant flux profile subject to the usual flux balance analysis constraints. This will give us important rate limiting or regulatory points in the metabolism that can be targeted for therapeutic purposes. Another approach that I'll take is finding the synthetic lethal targets, which are essentially a combination of multiple gene knockouts that restricts biomass production by blocking precursor synthesis. So to quickly recap, I tried to convince you that a highly curated metabolic model is essential and how experimental data can reinforce the predictive capability of metabolic models. I showed how specific regulatory points in metabolism helps understand the adaptability of mutants, and then what we can do to find higher order gene knockouts that can be promising to combat antibiotic resistance. All these works were performed under the guidance of Professor Saha in the Systems and Synthetic Biology Laboratory. Here are the social links for our group, and in this picture you can see the group members this summer, and we are all socially distancing. The funding sources for this work came from the Nebraska System Science Initiative and the University of Nebraska Collaboration Initiative grants. Thank you for listening to my talk. If you have any question, I will be happy to answer. You can use the chat box on the right hand side or use the Zoom link to directly talk to me during this conference. Thank you.